Hello everyone, and hope that you're doing well. Now today we're reviewing this CSEC/CSEC past paper for biology, paper 2 from the year 2022. Before I start this review, I want to point out a few things. One is that you need to read through the instructions carefully to ensure that you understand all instructions. The next thing I want to point out is that this is a structured paper. Therefore, it is about quality writing versus quantity writing. In other words, you will not necessarily get marks for the amount you write, but you will get marks for the valid points that you actually write for each question. It is therefore important then to use the number of marks to gauge your answers. It's also very important to read through each question to understand exactly what they want you to write. With that being said, let us start with our first question from section A. And the question reads that germination involves the breakdown of food stores in a seed. Figure 1 shows the results of an experiment where 50 germinating seeds were exposed to varying concentrations of pollutants in water. So what I want to do is to observe this graph a little bit. And so the graph is showing on the y-axis the number of germinated seeds. On the x-axis, it is showing the concentration of polluted water. And I want to notice something here that they actually start at 0% pollutant, which is distilled water, and it goes all the way to 100% polluted. And if you notice decline in germination going down at 100%, there is no germination at all. 0% and 1% is pretty much the same. I mean, 1% is slightly lower than the 0. All right, but they're pretty much the same. All right, and so let us now jump into our questions based upon this graph. And so for part A, it's a list 4 factors other than pollution that can affect the germination of seeds. And so we have temperature, oxygen, pH, and water. I want to point out something that in terms of temperature, some seeds require lower temperatures, while other seeds may require higher temperatures to germinate. So there's not one temperature that fits all seeds, all right? Oxygen must be available, and oxygen is available to at least produce energy, and energy is required for all those cellular activities that is required for germination. pH is also important because some seeds as well require lower pH, while other seeds require higher pH to germinate. So the pH must be right. Most seeds will prefer a neutral pH, all right? Water must be available, or moisture must be available. All right, so those things are the 4 factors that are needed for germination to occur. For part B, it says state the aim of the experiment. And the aim of this experiment, a suitable aim, a matter of fact, is to determine how the level of pollutant concentration in water affects the germination of seeds. All right, so anywhere in that line, you will be correct. All right, so for part C, it said using figure 1, explain how the concentration of polluted water affects germination at each of the following concentration. And so at 1%, it said there is little to no effect on the germination of the seeds. If you'll notice the graph, it's pretty much the same as 0. It said almost all of the seeds, all of the 50 seeds germinated. The pollutant concentration was low, hence there was no effect on the chemical or the physical processes of germination. All right, so low pollutant or low pollution really did not affect the seed at all. All right, let's jump into the 50%. For the 50%, if you notice the graph, it was, let's go back to one little quick view on the graph to show you, so you at least will see why the answers are that. So 50%, if you notice it, it's about 20 seeds that were germinated. All right, so this will be the answer now. All right, and this will be the answer here at 50%. Right, so at 50%, the germination is low. All right. Again, the germination might have been low due to high concentration of pollutant affecting the chemical processes of germination. And a point to note here is that at the 50% pollutant, only 20% of the seeds actually germinated, which is way less than half, right? And so therefore, in terms of this, you have 80% of the seed that did not germinate. And 80% loss is a big loss in terms of seed germination. That's really poor, all right? Now for part D, it said here that the research team, matter of fact, provided the results of the experiment to a farmer. State 2 ways in which a farmer can use the data to improve his farming practices. And so I have a number of things listed here. Some of them will be overlap, matter of fact, but I just want to point out the ways that you actually could answer this question. And so again, you don't need to write in long, extended paragraphs and epistles, really, just to make the points clear in terms of what you want to point out. And again, it's a 2 mark. That's why I asked for 2 ways. But I gave you 6. Again, some of them are overlapping. So one is to improve the seed germination by reducing contamination or pollution of water on the farm. So once you reduce pollution or contamination of water, you should have a higher germination rate based on what the graph is showing. You can also use the data to predict the outcome of the percentage germination based on the type of water used on the farm. So if you know it, if your farmer knows that they have a polluted water, then definitely it will kind of predict how much seed you'll get out of it. And so as a result of this, the third point, it can maximize yield based on the number of seeds sowed versus actual number of seedlings needed for planting. So in other words, if you know that you have 100 seeds, for example, right? And if he knows that his water is 50% polluted, then he knows he's only going to get 20 seeds that will be germinated. So you know that 80 seeds will be lost. So he can use the numbers to kind of predict how many seeds he actually will get. And so if you need higher number of seeds, then you have to plant much more seeds to get those. Of course, there'll be a lot of wastage, but at least you understand exactly what to do. Now, for the other points, which will be on the left right here, you can reduce seed wastage. And again, this is overlapping with number 3, but this is a way out to kind of answer the question precisely. You can also get healthier seedlings by reducing pollution, all right, pollution of the water. You can also get faster or higher germination rate based on using pure, purified, or distilled water, all right? And say on the graph here now, which is part C, so on the graph or on the figure 1, clearly place an X on the bar that represents the result of the control. And I placed that, I don't need to go back up there, but it was placed on the bar with the 0%, which is the distilled water. And part 2 now of part C is asking you to give one reason for your answer in C1. And the reason for this is because there was no pollutant present in that experiment. Hence, it can be used to compare the other results as a standard, or what they call a control, of what would have happened under normal conditions. In other words, without no pollutant at all. So that's what you're going to use to compare results to say, okay, if there was no pollutant, then this will be that. All right, so a control is always under the most normal or suitable conditions as possible. All right, so for part F of this, all right, it said that Mina investigated the growth of some seedlings and made comparison with growth patterns in a human baby over a 30 days period. The data are provided in table 1. And so table 1 we have here, we have from day 5 all the way to day 30. We have the height of seedling starting from 10 all the way to 30. And then we have the length of baby from 45 all the way to 64. All right, and so again, just observe a chart and then you will understand the answers that I'll provide. And so on the grid provided on page 7, plot the data obtained for both seedling and the baby using the same scales and axes. Provide a suitable title for your graph. So we're going to run through that, and that's a 6-mark question. And so we have the graph here. So matter of fact, let's go to the title first, so I don't need to go back to it. So the title of this is saying graph showing the comparison of the height of seedling and the length of baby over 30 days. All right, so that will be our title. And so for the graph now, for you to see everything, I need to decrease this size. So let's just decrease the size real quick. All right, so this will be our graph now displayed. All right, so you see a little bit better, more of the graph at once. So the graph should look something like this in terms of its shape. All right, so notice there we'll start from 0 going all the way up. All right, and so notice the height of seedling is always lower than the height of the baby. So this is the length of the baby in blue. All right, so that's the general idea for your graph. Should look fine lines, and your points must be clearly identified. You also should label the axes as well. So on my X, I put time in days. All right. And then on the Y, I put here the height or length in centimeters based upon the table. All right. And so once the graph is generally looking like that, you're getting those marks. So draw the graph properly to plot the points and also labeling of the axes and so on, and the title as well. All right, so that will account for your 6 marks. All right, so let's now jump back into the questions, and we are going to answer now part G. All right, and so for part G, is a suggest part G1, a matter of fact, is a suggest 2 ways in which the growth of the baby differs from the growth of the seedlings. And 2 points that I make right here is that one, the baby's length, right, are always higher than the heights of the seedling for the same time period. So that's why the baby's graph, notice in blue, was always higher than the one in red, which is for the height of the seedlings. So it's always higher in terms of the number. All right, and so for the second point is that the average incremental change of the height of seedling is more than that of the baby. So even though it was lower, the incremental change, which means from one point to the next, it always have a average higher increase. All right. All right. So in other word, what will show is that that seedlings will grow faster compared to the human baby. All right. However, not because you're bigger mean that you're growing faster. All right. So the seedling will have a faster growth rate for the same time period. All right, so part 2 now suggests one similarity between the growth of the baby and the growth of the seedling. And of course, they both increase with time. So that's a similarity in terms of what is happening. They both increase with time. All right, for part H, it's a describe how the seedling's height is measured using a ruler. And you measure the height by simply placing a centimeter ruler against the seedling with the 0 mark on the surface of the soil, because you're measuring from the top of the soil all the way to the top of the seedling. All right, that's a 1 mark question. All right, for I, is a suggest one example that shows that an increase in cells is not always due to growth. All right, for this question, it can be answered pretty much in different ways, because the way the question is stated, right? I want to point this out. They said, state one example that shows an increase in cells. This is not kind of specific, to be honest with you, kind of vague, because when they said increase in cells, do they mean increase in the size of a cell or increase in number of cells? Because there are 2 ways you can have cell increase. All right, so I want to show you how you actually could answer this question. So 1, you can put binary fission. 2, we can put meiosis. All right, and so for these 2, the number of cells will increase. So in other words, you have more cells, but you will not have more organism. All right? So you have more cells. So meiosis, remember, meiosis used to produce gametes. Okay, and that means you have more cells in the body, but that's not growth. Binary fission, for example, in bacteria, the cells will divide and produce new cells, which means new organism, but the organism is not growing, or the original organism is not necessarily growing. All right, you still have one bacterium that actually will produce more bacteria. All right. The other ones that I actually have here is water entering the cell by osmosis. You have an increase in temperature pressure in the cell, and that could be because of again osmosis or fluid built up. You can have storage of nutrients such as fats, carbohydrates, and amino acids. So when food stored in cells, then the cells get larger as well. And one example of that is when you're doing here layering and we have translocation of food into different parts of the stem, then you have bulging taking place. And bulging is because of the storage of food, and also in root tubers as well. The food may store as carbohydrates, as starch, and so on. And in this case, the cells increase in size, but not in the number of cells. So there are 2 ways in which you can have larger cells or increase in cells. 1 in terms of number and 1 in terms of size. All right. All right. So again, just to make those 2 points really clear. And again, I really want to be very specific when answering these questions. Again, we just need to understand what the question is really asking you, right? All right. So, but for this one, just to point it out before I go, it said that increase in cell though, I think that is referring to more likely the number of cells. All right, and so um, binary fission and meiosis will be the two most suitable. But again, in cell size, again, these options will be also important for increase in size. So you may not know what they may ask you based on that examination here, right? Because of course they will not ask you the exact same thing all the time. All right, so for part J, is a suggest one factor that should be considered before making a conclusion from the data in the table. And so things that we can look at in terms of making a conclusion versus length and height of baby is that we can consider the starting length or the starting height. Because of course the ceiling will be very small versus a newborn baby will be larger in size or length, right? Naturally. Also, we can look at the nutrient requirement or the difference in needs of nutrients, because a baby may require a lot of nutrients, and so you have to feed the baby much more in terms of quantity versus a ceiling. And also, again, we can look at the time during the day when the data were collected. It was after feeding, after watering, because of course, when you water plants, they be more turgid and more upright versus on hot days when they don't water, then therefore they're going to be more flaccid and wilt, and so they're going to look much shorter as well, right? So there are a number of things that you have to consider, the environmental factors and all those things, right? All right, so that's our first question, 25 marks. All right, so let's jump into the next question here, which is question 2, and it said define each of the following terms. And so the first one, we're looking at saprophytic nutrition, and these are organisms that are saprophytes. And so a saprophytic nutrition is the obtaining of nutrients from dead and decaying organisms or organic matter. All right. And so here now, heterotrophic nutrition, all right, which means heterotrophs, all right, which example us. And so when we talk about heterotrophic nutrition, you're talking about obtaining nutrients from other organisms. And so I don't make a point right here is that these organisms will intake and break down of organic substances. So they're taking substances or nutrients or food and break them down into smaller portions that can be absorbed. All right, that's what heterotrophs do. Now, autotrophic nutrition, which represents autotrophs, such as plants, they produce their own food or nutrients. So in other words, what autotrophic means, they are self-feeders. Auto means self and trophic means to feed. So they are self-feeders. And so they use inorganic substances with either light or chemical energy to synthesize food. And so I just want to make a note right here that light, those will be phototrophs. And if they use chemicals, they will be chemotrophs. All right, just to make that distinction between the different types of autotrophs. All right, so for part B of this question, it said complete table 2, identifying 2 saprophytes and their food sources. And so I list 4 right here, just for your information. So we have mushroom, we have yeast, we have some bacteria, and we have also have mucor or mold. And for mushroom and yeast, they are fungi. All right, and so they will eat dead plant materials. All right, and yeast, they will use sugary materials such as ripe fruits. All right, and that's where you get fermentation, fermentation to produce alcohol or wine. All right, and so bacteria now they will feed on dead plant materials. For those that are saprophyte, not all bacteria are saprophyte. Just to make a note of that, right? For mucor or mold, they will feed on carbohydrate-rich food such as stale bread. All right, so that's why you see those green stuff sometimes on bread when the bread becomes stale. Those are mold. All right, particularly pin mold or mucor. All right, for part C, it said explain 2 processes by which the raw materials required for making nutrients reach the leaf of a plant. And so to make the nutrients needed, the raw materials needed to reach a plant, to make the nutrient, one is by transpiration. And what transpiration does is to make water reach to the leaves from the roots. And so this is the detail right here how it actually occurred, that what is being pulled from the soil by the root hair cells through the process of osmosis. All right, and so due to capillary action, now the water molecules will climb into the narrow xylem vessels. All right, as temperature increase, then water molecules will evaporate, creating what they call a continuous pull, which we call a transpiration pull. All right, on the other water molecules will then result as a chain reaction, all right, or a continuous stream that we call it, a stream of pulling of water from the soil through the leaves. All right, for the next one here is diffusion. So diffusion next process, and diffusion actually makes carbon dioxide available in the leaf. And so when the guard cells become turgid, they result in the opening of the stomata, then carbon dioxide from the atmosphere, then diffuse from the higher concentration on the outside to a lower concentration, which is in the inside of the leaf. All right, so carbon dioxide is made available by diffusion. Now, radiation is under process here, and they ask you for two. I give you the three that are required. And so radiation is under the process, and radiation is a process by which sunlight travels from the sun into the leaf, and this is done in form of rays or light waves. All right, and so it states that when light rays hit the plant leaves in particular, they travel inside and then they are absorbed by the chlorophyll. And just to make a point that the small pockets of energy within light, they are called photons. All right, and so the photons now they'll kick the electrons out of the chlorophyll and to produce the energy needed to make glucose. Now for part D, it said young seedlings use all the nutrients stored in the cotyledons to start their growth. Explain the process by which the seedlings make more nutrients to continue their growth. And so this process is photosynthesis, right? Because once germination has ended, because of all the nutrients have been used up, then the plant will now start to photosynthesize. The germinating seed will start to photosynthesize from those first leaves. So the point here, I give it in point format, right? To make it a little bit easy and clear. So one is that once growth start taking place, the seedling will start to have their first leaves. All right. And so the second thing will happen here is that the seedlings start to make food by photosynthesis. And then sunlight is absorbed by the chlorophyll in the leaves. What is absorbed by the roots or the radicals? And then they are sent to, so this is supposed to be sent here, and then are sent to the leaves through the xylem. All right, and then what will happen here is that carbon dioxide is taken into the leaves through the stomata, and then we have light is being absorbed, and the light that is absorbed will split water molecules, producing hydrogen ions and oxygen gas. Oxygen is a waste, but the hydrogen ions will be used now to react with carbon dioxide. All right, I have it right here. So hydrogen ions react with carbon dioxide to produce glucose. Okay, so react with carbon dioxide to produce glucose. All right, and glucose is a sugar. All right, just to make that point. All right, so let's jump into the next part of the question, or yeah, this is the next question actually. So question number 3 is a figure 2 below shows a typical plant cell and a typical animal cell, as you will see them on the labelings. And the first thing they ask us to identify the structures labeled A, B, C, and D. All right, so A, let's go back up a little bit. So A, if you notice, A is the outer part of the animal cell and the second layer in the plant cell. And so that will be our cell membrane, otherwise called our plasma membrane. B is the cell wall. All right, it's not on the animal cell, it's only on the plant cell. C is the cytoplasm, and D is the vacuole. Okay, and I want to point out something here. Notice the plant cell has one large central vacuole, while the animal cell will have small vacuoles scattered within the cytoplasm. All right, so we identified that. And of course, there are other points, other structures that you need to identify as well, such as the mitochondria, which is right here, the nucleus. Also, we need to identify the ER, the rough and the smooth, if they should show you those. All right, ribosomes and so on. All right, so for part 2 of this, it says state the function of any 3 of the structures identified in A1. So we're going to state the function of any 3, but again, I give you all of them that were identified here. So the cell membrane controls what enters and leaves the cell, And of course, I know that cell structure should be easy for most of you, if not all of you. All right, now cell wall maintains the shape of the cell. Cytoplasm holds the organelles in place and the site for most chemical reactions. Vacuole stores water, nutrients, and also waste materials. All right, so let's jump over that. That one is a relatively easy question and straightforward. Now for part B, it's a student collected two specimens, specimen A and specimen B, from a seashore during a biology field trip. She used a microscope to determine which structures were present in the specimens and found that specimen A contains more mitochondria than specimen B. And here's asking us to suggest two reasons why specimen A had more mitochondria than specimen B. All right, and so there are a number of reasons why this may be possible. Again, state and explain is very important. So if you state two reasons and explain them, then that accounts for your 4 marks, right? But I list 4 here and explain them. So this is a total of 8 marks. But again, you list 2 and explain them as they actually say 2 reasons. Again, use your marks to gauge your answers. Now, one is that specimen A may move more than specimen B, naturally because if you move more, then you require more energy. And if you require more energy, naturally the cells would adapt to have more mitochondria. All right, so hence a greater number of mitochondria in specimen A because it's more motile. All right, so specimen B may be not a motile organism, may stick to one place, and not moving really. Now for part 2, or the second option here, is that specimen A may be larger than specimen B, and larger cells or larger organisms tend to carry out more cellular activities. Hence, they have more cellular activities require more what? Energy, because most cellular activities require energy to take place. All right. Now, for the third option here, you have more frequent reproduction or growth. So if specimen A is growing more frequently or more rapidly or reproducing more, but these could be 2 separate points. Reproduction and growth could be 2 separate points. And so any of these, they require energy for the cell to divide, right? So growth and reproduction requires cell division, so therefore require more energy for cell division, of course. For the fourth option, specimen A could be a carnivore. In other words, carnivores tend to hunt their food, all right? And so if you're hunting versus just simply absorbing what is around the environment, then naturally you require more energy to move fast, to send out probably stinging cells, to do movement and all those things. So definitely carnivores tend to require more energy versus herbivores. All right. Now part C is using 2 named examples, explain why cell specialization is important for cells to function effectively. And I want to make a point here in red. It said that what this is actually explaining or defining what cell specialization is. And cell specialization is when cells transform to perform specific functions. All right, so it is a transforming of cells to perform specific functions. So the name examples here will be 1, I have sperm cells, and sperm cells now, they are specialized by developing what they call flagella, all right, which enables them to swim effectively through the female's reproductive system. You have ciliated cells, for example, and so there are many cells you can talk about. You have ciliated cells, and ciliated cells, you may find them in the nasal cavity. You may also find them in the trachea, and they develop what they call cilia to sweep unwanted substances out of the breathing system. All right? And other ciliated cells, the cilia can also assist with movement as well because they're similar to the flagellum. Muscle cells, for example, they are adapted to contract and relax to bring about movement. All right. So again, you can name so many different types of cells. You have cells. Well, I'm not talking about the cell that I don't list here. You have those cells that are phagocytes. All right. So phagocytes now are white blood cells in our body that will have that abnormally shaped nucleus. And the reason for that abnormal shaped nucleus is also to facilitate the different induced shape of the cells. So here, the phagocytes, which are part of our white blood cells, they adapted to induce specific shape to engulf microbes by what they call phagocytosis. Right? We also have nerve cells or neurons, and they're adapted to transmit electrical messages around the body. And these electrical messages, they are called impulses. And so they are insulated by a structure called the myelin sheath. All right. And so just like electrical wires, they are insulated. So different cells will do different things. We have cells that are in our taste buds. All right. And those cells will adapt to detect chemical taste. We have those that in our olfactory, which are in our nostril, that will detect the chemical for smells. And so we have so many different cells in our body that you can list right here that adapted to do different things. Now, question number 4. Let's kind of speed up a little bit. And it said define each of the following terms. And so we have climate change, and climate change is the long-term changes in factors such as temperature that affect climatic conditions. We have population, which is the next option here. And population is the total number of the same species. You must remember that. The same species that live and interact within the same area at the same time. That's what a population is. All right, the same species, very important. Without that, you're going to get it wrong. I can guarantee you that. Part B of this question is a state 4 methods that may be used to conserve the environment. And so there are so many different methods you can use. I think you could just run home with this one by listing many. And I know most of you will know these things, right? So to conserve the environment, you can reduce, reuse, and recycle. That's most popular and the one that most of you remember. We can also do reforestation, planting of trees or replanting of trees. We can use renewable energy instead of non-renewable energy. We can reduce the use of non-biodegradable materials. We can design areas as protected areas. Well, designate areas as protected areas such as parks, national parks, and so on, right? We can do all of that. We can implement and reinforce open and closed season for specific organisms. All right, so you not hunt them at a certain time, you not capture them for food at a certain time. You give them a chance to reproduce. You can reduce the use of automobiles or by doing carpooling and things like that. You can also convert cars into electrical cars or using specific type of fuel, which are renewable fuels such as alcohol. We can practice good solid waste management and sewage waste disposal. And so we have many more things we can talk about. But again, you can run home with this one. All right, for the next part of question here, it said that the Earth climate has warmed considerably over the past few decades. So this considerable increase in temperature will be very important. They say using 3 examples, explain how global warming may lead to an increase in infectious diseases within the Caribbean region. And so, point to note about infectious diseases is that infectious diseases they are diseases that are caused by pathogens that enter the body. So once pathogens enter the body, that's called infectious diseases, right? And so an increase in temperature to a considerable amount, these are some of the possibility. Because I'm using 3 examples. So I look, dengue fever is one. And just to give some additional information as I go along, it's more than what is required, to be honest with you. But just to give you a kind of brief overview of the entire examples, I just put some extra notes in. All right, and so for dengue fever, which is a viral infection, right, it's caused by dengue virus, and it's transmitted to humans through the bite of infected Aedes aegypti mosquitoes. All right, and it said this may increase due to the global warming, because global warming, what that does is that warmer temperatures may increase the time which these mosquitoes reproduce. They have longer reproduction periods, all right? And mosquitoes will love that. So therefore, more reproduction period or longer reproduction period will increase the number of mosquitoes, hence increase the possibility of dengue fever. The chikungunya, for example. Again, this is also a viral infection transmitted to human through the bite of infected Aedes aegypti or the Allopictus mosquito. All right, so the Albopictus is another version of mosquito that really can cause chikungunya, but the most common one, again, is the Aedes aegypti. All right, it said this may increase due to global warming because warmer temperature may increase the time which these mosquitoes reproduce again, just as dengue fever. Again, these are overlapping information, but again, they say list two explain both of them. And for the third option here that you can have here is Zika virus. Again, this is also a virus, a viral infection transmitted to human through the bite of the infected Aedes aegypti or also the albopictus mosquitoes. Again, if you notice that they are similar. So the first three, they're pretty much similar in terms of the vector that is causing it, all right, and the effect of the increase in temperature. So the increase in temperature, again, will also cause increase in time of reproduction of these mosquitoes. Cholera, which is kind of different from the from the first three. So the first three could list any of those and then go down to cholera. So for cholera, it is caused by consuming contaminated water. So it's a bacterial infection caused by the Vibrio cholerae bacteria. All right. And it said, even though it is caused by contaminated water, warmer temperatures may increase the replication of the bacterial cells. So bacteria may replicate more in warmer temperatures, right? Therefore, increasing the number of cells per volume and the probability of infection. So more bacteria per volume of water may give you a higher chance of infection. Not only that, because water is moving more rapidly on warmer days, then the bacteria may spread to a far degree, which I'm going to explain at the end. I think I have some notes there as well. For the fifth option, you have malaria. And malaria is caused by a parasite transmitted to humans through the bite of infected female Anopheles mosquito. All right. And it's a warmer temperatures may also increase the reproduction and spread of these parasites. All right. And the sixth could be like the coronavirus, the flu, and any airborne diseases. And it's simply put because warmer temperatures may increase faster movement and activities of most pathogens that cause airborne diseases. Because remember now, gases will move faster. So anything that is airborne or attached to gas molecules or air or water molecules in the atmosphere, warmer temperatures cause them to move faster and spread at a far further space or take up more space. And so therefore higher chance of infection. Some points here to discuss. When you're discussing these points, I just list them there instead of explaining them because I don't have enough space to write them. But what you can also explain or interject into all of those up top is that global warming causes changes in rainfall, storms, flood, and extreme events. And global warming causes these changes. And if you have more flood, more rainfall, and more extreme events, then what they can do is to spread these vectors from place to place. For example, if you have flooding start at one place and the water will continue to another place, so therefore it can transmit like bacteria or transmit egg cells from mosquitoes and so on. So there are so many things that these things can cause and more spread of these diseases. And also flooding could also lead to high contamination of the water as well. So we have increased contamination of food and water due to global warming. We have increased pathogen and their activities. Those are points you can interject in those explanations. You can have expansion of infected area. Again, this could due to flooding, storms, high wind, and all those things could really cause the expansion of the areas that are infested with mosquito or even with the any vector, matter of fact, including bacteria. All right, let's jump into part D. There is a climate change can severely impact small island developing states, which are called SIDS, within the Caribbean. Suggests three possible actions that can be taken to reduce the effects of climate change. And so some of the things that we can do here to reduce climate change is one is to invest in electrical cars to reduce the dependency on fossil fuel. We can also invest in renewable energy, and we encourage individuals to participate in renewable energy practices or projects. We can encourage and promote tree planting efforts. We can encourage coastal cleanups and proper waste disposals. We can also ban the importation of and use of specific chemicals such as pesticides, commercial fertilizers, aerosols, and corrosive substances. We can place sanctions on the proper disposal of derelict vehicles. Derelict vehicles, those are vehicles that are, you call them not usable or not functional vehicles that may park in yards, and those can cause infestation of pests and rodents and so on, and even leakage of chemicals in the soil as well. So derelict vehicles are vehicles that you should not really have hanging around. The proper disposal of those, they can collect them, a matter of fact, they can recycle them, they can use parts out of them, and things like that. All right, you can encourage backyard farming and other sustainable efforts. All right, so those are some of the few things that you can actually talk about. All right, so question number 5, we're getting close to the end now. All right, so for part A, for question number 5, it says Ellis received a blood test from his doctor. His test results show that his white blood cells and platelets level were normal, while his red blood cells level were not normal. Complete table 3 below to show one function of each of the blood components. And so quickly we're going to run through this. Red blood cells, they transport oxygen to the cells of the body, and they also take away carbon dioxide, just to make mention of that too. White blood cells, they help to protect the body from pathogens or diseases. And platelets, platelets aid with blood clotting to prevent blood loss when they get injury or a cut. All right, so into part 2 of this, it states that name 3 other substances which are found in the blood. And so I answer this in 2 ways. So let's start with the answers in blue first. So all of these substances are in blood other than the blood cells, because what is in the chart, red blood cell, white blood cell, and platelets, they are blood cells really. So beside the blood cells, you may have plasma, you have hemoglobin, you have oxygen, you have carbon dioxide, you have amino acid, you have hormones, you get the drift. You have glucose, you have ions, you have antibodies, you have water, you have urea. And the next way to answer this in terms of category, you can do it in this way as well. You can say nutrients. And nutrients include the amino acids and so on, the glucose and all of that, right? The gases will include the carbon dioxide and oxygen. Hormones, and hormones, of course, they are proteins, so that includes the proteins as well. And even other blood proteins included here. And so we have waste, we have electrolytes. Electrolytes will include ions, and different form of ions, such as potassium or calcium and so on, calcium ions. We have proteins. Hemoglobin is a protein. Hormones are proteins. Antibodies, of course, that are produced from the white blood cells. All right, so those are the category. You can answer this question in terms of answering a categorical form, or just specify the specific or the exact substances. So it really don't matter, to be honest, the way you answer it, because it's a open question. It's a name 3 other substances which are found in blood. All right, so you can choose to answer any of those 2 ways. All right, let's go to part B now. And for part B, Ellis received additional test which revealed that he has sickle cell. Oh my. And this is sickle cell anemia. All right, so sickle cell anemia here is suggests one way in which Ellis red blood cells may differ from a person who does not have a sickle cell anemia. All right, so sickle cell is a very distinct disease, right? It's a genetic disease, just to mention that, to have a cell sickle. And so it'll be different in terms of, one is that it become a crescent shape instead of a really nice biconcave shape. And then you have the red blood cell will get stuck, and they reduce blood flow and deprive the cells of oxygen. And before I even go to the second point, I want to make a note right here, right? Anemia can be different from anemia is different from the sickle cell anemia, right? That's what I want to point that out. Because you have anemia, which is just a deficiency disease, which is the lack of iron or lack of hemoglobin. But you talk about sickle cell anemia now, you're talking about the abnormal shape of the red blood cell. And once it's abnormal, then of course blood flow will reduce, cell will be deprived of oxygen and nutrients and so on. So the cells will also be less flexible. And because they're less flexible, then movement will be reduced, and also flow will also be reduced. They become more sticky. And because they're more sticky, they reduce movement or flow as well. All right? So let's now jump to the next part of the question. Now, Ellis becomes concerned that his child may also develop sickle cell anemia. His doctor tested his wife's blood and told Ellis that it is unlikely that he will have a child with sickle cell anemia. So unlikely, will not have. All right. It said with the use of genetic diagram, explain why this is not possible for Ellis to have a child with sickle cell anemia. In your answer, let S denote the allele for sickle cell trait and A the allele for a normal trait. All right, so they give you two different letters here: S for sickle cell and A for normal allele. And so the phenotype of a father, which is Ellis, is sickle cell. The phenotype of a mother, she has to be normal, because that's the reason why Ellis' child or children will not have sickle cell anemia. The mother must be normal. But I want to point out something here in terms of genotype of the father, because he's sickle cell, and let's consider sickle cell as a recessive disease, recessive trait disease, then he must have both alleles for that disease. For the mother who is normal, the mother could have been two things. She could have been a carrier, or she could be a homozygous normal. But I'm choosing homozygous normal. Why? Because if she's a carrier, then she can give one of those sickle cell allele to Ellis' child, and then that child will have sickle cell. But in order to prevent that, she cannot have a sickle cell allele. She must have only normal allele, which means one of her allele must go to each child. And since the normal will be dominant over sickle cell, then one of the dominant allele will indicate that there will be no sickle cell at all. At least one dominant allele, no sickle cell. And so the genetic cross would be this with our Punnett square. All right, so if you notice, I put the mother here on top of the chart, and the side of the chart will be Ellis. And so all of them, the result will be AS, which means that all of them are normal because they have at least one of the dominant allele. All right, so the explanation here now is that all the offspring will have a copy of the allele of the normal blood. And so since sickle cell is a recessive trait, then all of the offspring will have a normal red blood cell. However, a point to note is that all of the offspring will be sickle cell carriers. So they are carriers, so their phenotype will be normal, but they are carriers in terms of their genotype. Okay, so they are heterozygous or hybrid, in other words. All right, so hybrid or heterozygous, they will be carriers. All right, that's 15 marks. So we finish with that, and we are on our 6th question. Now, and this will be our last question. So Anna has a 28-day menstrual cycle. Figure 3 shows the changes which occur in Anna's uterine lining during her menstrual cycle. And the uterine lining right here is called the endometrium. So this is the endometrium that is shown here. Notice at these days, the 3 and 4 and all the way to 6 and 7, it's really a decline and it starts to increase. So this is where menstruation actually takes place. Day 14 is where ovulation, approximate ovulation point. All right. And so after ovulation, going up, the uterine lining is much more thicker. All right. And it said here now, we should do this in part A1. It said name the 2 sex hormones which are associated with the menstrual cycle. And these 2 hormones are estrogen and progesterone. Of course, you know, the secretion of these hormones are also contributed or affected by the FSH and also the LH as well, all right, that are produced by the pituitary gland, all right? But we're not getting to that now. Maybe a next exam review we'll talk about those. All right, so it said on figure 3, sketch a line for each hormone to show how the levels of the sex hormone named in A1 change over the 28-day cycle. Label each line. And so labeling them, if you notice that estrogen was a little higher than progesterone at the first part of the cycle. However, leading up to day 14, where ovulation is going to take place, estrogen will surge and then it start to decline after that. And after ovulation, then progesterone will be at a peak. So notice slightly start to increase before ovulation, however, but after ovulation it becomes peak, and then after they start to decline especially if there's no fertilization of an egg. Notice again, after the 28 days period, estrogen will start to get a little bit higher than the progesterone, just to make a note of that, while progesterone start to get a little bit lower than estrogen. All right, so let's now go into the next part of question. No, well, yeah, next part of question. And this part reads now, which is part B. It said that Anna recently became pregnant, and I hope she's married. It's explain the effects that pregnancy will have on the two sex hormones named in 1A. All right, sorry, in A1 actually. And this here now, to answer it, estrogen, just to make quick notes, estrogen gradually increased during pregnancy until it peaks. So that's what generally is happening to estrogen. If she becomes pregnant, it starts to gradually increase and then it peaks and then it will tapers off or plateaus in a sense. All right. And so what is the purpose of this though, just to make some extra notes here about why estrogen will do that, is simple to cause an increase in blood flow to the uterus, and more blood flow to the uterus, which means the uterus will have more nutrients and oxygen that is needed for the development of the embryo and into a fetus. All right, so blood is required for all of that development, for the cell division to take place to turn the embryo into a fetus. And also, what you have here is also an enlargement of the uterus. And once the uterus is enlarged, it's really to accommodate the developing fetus. And also, it helps to maintain the uterine lining by supplying the nutrients and other things that is needed for that development of the uterus. It also stimulates the mammary gland development and also enlargement of the mammary glands. The mammary glands are what the tissues that are in the breast that will produce milk. So those glands, they start to develop more. All right. And so the estrogen also stimulates the secretion of prolactin, and prolactin is needed for breast growth and also milk production. All right. Estrogen also relaxes the pelvic ligament, because the pelvic ligament must relax to accommodate that expansion of the uterus, and also ease on pressure, to reduce pressure on the uterus itself and also on the vaginal passage. All right, because higher pressure will cause more stress on the urinary bladder, also in the uterus, and that if that takes place, then you can also have some problems. Also, what estrogen does is also to prompt uterine contractions, but this uterine contraction is not in or during the period of pregnancies until the ending of pregnancy, when you want to contract the uterus to cause the amniotic sac to break and then start to release the child for birth. All right, so it is not during all the stage of pregnancy, it's at the end of pregnancy, almost the end. Then you have that stimulant or stimulation of the contraction of the uterus. Now for progesterone, progesterone does the same thing, gradually increase during pregnancy until it peaks, or it will plateaus when it is maximum. And so the purpose for progesterone doing that is really to prepare the endometrium for implantation, right? That is the early part of pregnancy. It also to maintains pregnancy, so the entire pregnancy maintains it. It also helps to enlarge the uterus, just as estrogen does. Prevents other eggs from maturing, because once you become pregnant now, it will prevent those eggs from maturing, to prevent menstruation, because if you start to menstruate, it can also decrease the wall of the uterus. And if you menstruate while you're pregnant also, it can cause miscarriage, all right? And so it prevents uterine contraction as well, which is relaxing of the muscle, and that also prevents miscarriage as well. Strengthen the muscles in preparation for labor, because the muscles now must become stronger. And this is at almost the end part of pregnancy, third trimester they're about. And when it become really, really strong, and the strengthening of the muscles, especially in the uterus and the pelvic area, is really to create that high pressure onto the amniotic sac, to create a force that's strong enough to expel the baby. All right, so she really needs strong muscles to do all of that. Now, for part C, it's after the delivery of the healthy baby boy, Anna decides that she does not want to have any other child at this time. She, however, wishes to have another child in the future. So notice that is very important. She decides to have another child or children in the future. So it said, recommend one suitable method, birth control method, which Anna could use to prevent pregnancy. And so she could use a number of things, and they only want one. So condoms are suitable ones. Pills, abstinence. Well, if she's married, then that will cause some problems. We're going to talk about that later, though. Injections, IUD, rhythm method, coitus interruptus, which is withdrawal. And this is very important. I put in red here. You cannot use tubal ligation. Why? Because tubal ligation is said to be irreversible. And if she wants to have children in the future, then it is not a suitable option. It is not recommended. All right. So they will mark you wrong if you put that on the paper. All right. Now for part 2, it says state 2 advantages and 1 disadvantages of the birth control method recommended in C1. So we want to talk about all of these. And so let's start with condoms. Condoms, well, the advantages are in blue, the disadvantages are in red. And so for condoms, they're readily available, prevent STDs. The disadvantage can burst. For some people, they have allergy or allergic reaction to latex. Reduces pleasure in some cases. And those are the disadvantages. Now, for pills and injections, they're effective, very effective indeed, but it does not affect intercourse, which is a good thing as well. But STDs are possible. All right. And another disadvantage is that prescription are needed, and you may have possible side effects as well. For abstinence, very effective, no cost, no STDs, totally safe, 100% effective, but may be difficult to maintain because of temptations. All right. And also, if the female is married, that can also cause domestic problems or conflicts because the lack of intimacy or lack of sexual intercourse in that marriage may cause problems. So it's not recommended for persons who are married or who live with someone, for sure. All right. So IUDs, they are very effective. They are long term. The problem with IUDs in terms of disadvantage, they can cause bleeding in some cases, and they do not prevent STDs. So STDs are possible. For rhythm method, and rhythm method is really to kind of study the menstrual cycle and know when is the safe days that you can have sexual intercourse without getting pregnant. Rhythm method is inexpensive, well, no cost to it, no side effects. However, very unreliable because you can have changes in hormone production or secretion of hormones, which can cause changes in ovulation, and then you may have a little bit of problems. Of course, it's not just as easy as said because you have to test the basal temperature of the female to test, temperature is right and all those type of different stuff. STD is possible. The withdrawal method, inexpensive, no cost to it, no side effects, but it's very unreliable, and STD is possible. All right. All right, so let's jump into part D. It said not all birth control method can prevent both pregnancy and infections from sexual transmitted diseases. And they say ask to state 2 sexual transmitted diseases, and we have HIV, we have herpes, we have gonorrhea, we have syphilis, we have chlamydia. And the next part of question here, we said identify a birth control method which would be effective at preventing pregnancy and also infection from sexual transmitted diseases. And this one is condoms for sure. It's a 15 mark, and it also brings us to the end of the examination. I want to thank you for watching this review, and I hope to see you soon with other reviews. Stay safe, stay blessed. Good luck on your examination.